Hello everyone, welcome to my YouTube channel. So let's discuss about the fifth module of Genomics, Proteomics and Bioinformatics. In this module we are having major three topics are there. One which speaks about detection functional sites in DNA, that means with respect to ORF, promoters, transcription factors binding sites, then translation initiation sites and all. And another one we will be discussing about the molecular modeling, the 3D modeling stuff. And the one more section is there, detection of functional sites with different tools for bioinformatic tools that we will discuss. Majorly three topics. Under three topics there are again subtopics are there, four topics, 5 topics, different subtopics are there that we will discuss. So in this detection of functional sites in the DNA, we are having four different subtopics that we will discuss in this video. So general approaches in gene predictions, there are two approaches are there. One is based on structural annotation, another one is functional annotation. Annotation is nothing but what? Assigning a function to a particular site, okay, or a particular gene. Okay, so a structural annotation can also be called as a gene identification, whereas functional annotation can also be called as assignment of gene functionality. That means what is the function of that gene. Okay, so structural annotation depending on the structure, right? So we are having open reading frames, right? We are having coding sequences, that is CDS, then gene structure is there, regulatory region is there. Wherever site, region, structure, all these things will come into picture, those automatically will become the based on the structural annotation, okay, in a DNA sequence using computational gene discovery algorithms and all. We will finalize it. So those who are dependent on the structures will come under structural annotation. Those who are, when you speak about functional, there has to be a specific function which is assigned to a particular gene, that is called gene functionality. It can be the biological function, biochemical, gene expression, regulation, interaction, to all whatever the predicted genes are there, using a sequence similarity search against other genes of the known function by using BLAST or FASTA. We have done it, right? Like in earlier classes and all, we discussed about multiple sequence alignment, all those things and all. So categories of gene prediction programs, there are majorly two categories are there. Now you know, based on the annotation, we are having a structural and functional. Whereas categories, we are having two major categories, that is ab initio based method, homology based method. All of you know ab initio method, usually it takes like speaks about like it will act as a template method. You compare with the known with the unknown. Whereas homology, you compare two things, okay, and find this how much similar they are. Okay. So let's start with the ab initio based approach. How these tools work based on this approach? It predicts genes based on the given sequence alone, only a single sequence. You take it and you predict few characteristics of it. It relies on two major features. How it will do? Depending on two major features. One is existence of gene signal. That means a particular gene has got a particular function, or it's it itself signals that it is something. Okay, when I have AUG, it means that it is a start codon. Understanding? When I have UAA, it means that it is a stop codon, right? These are all what gene signals, right? Depending on what they are meant to be, a particular signal we are having. Like for example, start codon, stop codon, intron splicing signals, transcription factor binding sites, okay, ribosomal binding sites and poly A site. These all are examples of existence of gene signals. Okay, so this is one of the major feature. Okay, based on that we are going to predict based on the ab initio method. Okay, here with the sequence alone we are going to see. Next, gene content. How much content is there? For example, statistical description of coding regions. Okay, depending on stats. Okay, this entire how much content it is, GC reach, that kind of stuff. Like ab initio method is also known as a template method. It identifies statistical patterns that distinguish coding regions from non-coding regions. We are having statistical patterns, what are there in coding regions one side, compare that with the non-coding regions. So you will get a probabilistic models like like you can use the probabilistic models like Markov model. Then Markov model or hidden Markov model, HMM. Okay, that can be used for identify for to get the data, the statistical patterns for both coding and non-coding sequences. And whereas homology based approach, what it will see, it will look for significant matches of a query sequence with the already sequence which are available in the database, like what you do in BLAST, same thing. Next, relies on finding coding regions by their homology to known expressed sequences. That means it completely depends on how much similar they are. Understanding? So these are the two approaches we came to know. The two approaches are there for structural annotation, based on annotation, structural annotation and functional annotation. Then categories, we are having two major categories for gene prediction program, ab initio and homology. Whereas ab initio, single thing will be there for coding and non-coding statistical information you take, and probabilistic models you build based on the hidden Markov model. Whereas homology, you compare two sequences and then you find the information regarding the unknown sequence, right? Next, so now this is about the introduction about how you are going to find the functional sites in the DNA. You have done the lab also, so hopefully it will be easier for you to understand. Next, predicting ORF. Under this, the first topic, subtopic is predicting ORF. What are ORFs? ORFs are nothing but open reading frames. What exactly they mean? They are nothing but a short, it will be, make sure that the ORFs are nothing but once it is it's like after transcription and translation, they are going to become a protein now, or you can say the polypeptide. Okay, the simplest method of finding DNA sequences that encode proteins is to search for ORF. If ORF is there, then that means it is going to form a protein sequence. Okay, so ORF corresponds to the stretch of DNA that could potentially be translated into the polypeptide. That means we are going to get the protein out of it. If at all the ORF sequence is small, you will not get the protein. So there are 6 possible reading frames are there in every sequence. A reading frame of a genetic sequence that does not encode a protein will have short ORFs. Imagine a particular DNA sequence is not coding for the protein, that means it has a short ORF. Okay, due to the presence of many in-frame stop codons. When you are having the short ORF, why you will have short ORF? Because many stop codons will be there in that sequence. When you have many stop codons, definitely it will not translate, because for translation to initiate, a start codon will be there, and as soon as it initiates, suddenly, since many stop codons are there, it will not grow. If the chain is not going, you are not going to get the polypeptide chain. So a stretch of DNA that would code for a protein, for you can define the ORF in this way also. A stretch of DNA that could go for a protein of at least 100 amino acid is considered as an ORF, which is a good candidate for coding cellular protein. So when you say there is a ORF, definitely that is somehow you can identify, you can say that okay, it is going to be translated to the protein. That's how you predict. Okay, so there are many tools are there. ORF finder is there, as its name itself indicates. Translate is there, ORF gene is there, GenScan is there for eukaryotes, rest all for prokaryotes. ORF finder, what it will do, it identifies 6 frame ORF. Input will be the FASTA format sequence as you do in NCBI. Next, you have to select the genetic code. Next, output will be the bar graph showing the ORFs, and a list of 6 ORF. Translation will be there because 6 reading frames should be there. Indicates stop codon also. Star, if it is there in the sequence, that it indicates there is a stop codon in the sequence. Next, translate tool at XPASI, that is also is there. Translate. Okay, second one. Now DNA sequence is translated into 6 frame protein sequence, that is 6 open reading frames. Various outputs will be there. There will be any nucleotide, amino acid, nucleotide with amino acid sequence. These 3 options will be there. Next, stop codons is indicated by hyphen. If hyphen is there in the translate, that indicates stop codon, whereas ORF it is a star. Next, ORF gene tool for web gene. Okay, we are having ORF gene tool that for web gene, that is here the query sequence should be in nucleotide sequence. It predicts exons and then search for the translated amino acid frames. When you speak about the exons, then there only you are talking about coding regions. Then they will search for what? The translated amino acid fragments. That is like indirectly you can say it as a protein sequence itself. And finally we are having GenScan, because here 4 tools were there, no? ORF finder, translate, ORF gene, now GenScan. Okay, now GenScan tool, it follows 2 approaches, both the approaches, ab initio it will follow, homology based also it will follow. So what it will do, it will combine both the information, like the information about genetic feature statistics with probabilistic model which you are getting in ab initio to predict the gene structure. Next, input genome sequence will be there, so you have to select the organism. Next, genomic genomic DNA is translated into all 6 frames, as we discussed in the previous slide. Next, that is translated exons are then it will be used to compare with the user supplied protein sequence, that is query already existing sequence. Okay. Next, translated genomic regions have high similarity at the protein level, receive higher scores. If the similarity is high, then you have a high score, right, like what you did in BLAST. Now the same sequence is again predicted with GenScan algorithm, which gives exon probability score. There is extra one thing is there in addition to the BLAST what you have done. What is that? Here GenScan algorithm will give you one number, that is exon probability score, what we call. So final exons are assigned based on the combined score information from both ab initio and homology based approaches. So once you get that, so you can say that the complete amount of DNA which is going to be translated into protein. Why we are behind of all the entire, what are the sequence which are converting into protein? Because the topic is prediction of functional sites. If you want to say that a particular genetic DNA sequence is functional, you have to get the protein out of it, understanding, that is the reason. Next, output include clickable view of gene structure of in a PDF or a PS images. Next, a table will be there will be image and there will be a table, and table as we saw in BLAST, similar kind of information we get here. Image there was there, here also image you will get, and here there was a table based on the maximum similarity. Here again listing predicted gene types will be there, initial, internal, terminal, exons, single exon, gene, promoter, all these different columns will be there, their information will be displayed, their lengths, beginning and end point, reading frame, score of the predicted features. If you just go and if you just recall your detection of functional sites experiment in the bioinformatics lab, you will understand. Next, table is followed by the amino acid sequence. After, just below the table, amino acid sequence will be there of the predicted translate, and nucleotide sequences of the predicted exons will be there. Okay, because both it will combine. So that was about GenScan. So that was about ORF, predicting ORFs, because predicting ORF itself is nothing but predicting the protein is going to happen now. So once that is happening, that means these so much genes are functional. So that is nothing but detecting functional sites in the DNA. Now ORF is done. Next we are having the promoter prediction. Similar fashion, I am not going to explain in detail again, because the same concept here, we know what is promoter. Same thing is mentioned in the introduction. Promoters are DNA elements which serves as a binding sites for the gene transcription machinery, then consisting of RNA polymerase and transcription factors. You know, these elements directly regulate gene expression. Also, you know, promoters and regulator elements are traditionally determined by the experimental analysis, but they are time consuming and laborious. That is the reason computational predictions are promising, because it has the potential to replace a great deal of extensive experimental analysis. These all we have studied as an introduction for bioinformatics lab or a bioinformatics module also. So, prokaryotic promoter prediction. So here operon prediction is there. So we are talking about promoter prediction. So operon prediction is nothing but it's a very key step in prokaryotic promoter prediction. Okay. So lack operon, all those things you might have studied, right? Because genes within an operon, what happens, they share a common promoter, right? When they share a common promoter located upstream of the first gene of the operon, and when you are going to detect that promoter, so by detecting, predicting the promoter, we are going to detect the DNA functions of the functional sites of the DNA sequence. There are two tools, BPROM, FindTerm. BPROM, most frequently used program compared to FindTerm. Web-based program for prediction of the bacterial promoters. We know that because that is under which topic we are discussing. It first predicts the given sequence of bacterial operon structure. It will go and it will find the operon structure of bacteria by using an intergenic distance. What is the distance here? Intergenic means from gene to gene. What is the distance? It has to be 100 base pairs as basis for distinguishing genes to be in an operon, because if it has to be in one single operon, 100 base pairs should be must. So once the operons are assigned, what it will do, what happens, it predicts putative promoter sequence. Okay, that means putative is nothing but like immature, you can say, those kind of it's like just pre-promoter sequence. Okay, understanding. So it makes use of ab initio approach of prediction. Okay, so here what is the approach? Ab initio, not homology. Next we are having FindTerm. FindTerm searches for bacterial RHO independent termination signals which are located at the end of the operon. You know that it is located in the end of the operon. So when you are going to find the bacterial RHO independent termination signals, you are going to find the operon. You are going to predict the operon. So predictions are made based on the matching known profiles, okay, of the termination signals which are combined with the energy calculations for derived RNA secondary structure, for again putative hairpin loop structures. So here with respect to the energy calculations, because here we are having the predictions, and the energy calculation happens whenever there is a termination signals. Okay, so once the termination signals are there, definitely energy calculation has to happen, because we are having the electron releasing and all those stuffs. So finally with respect to that, we are going to match the different profiles. So now the sequence regions that scores the best in features and as well as energy terms is chosen as the prediction. So with respect to the feature, with respect to the energy calculations, if these two things matches or their score is high with the comparable sequence, then that will be called as, the prediction is better there. This information is also useful for the defining an operon. So that was about the promoter prediction. So okay, so two tools, that is BPROM and FindTerm. Next we are having eukaryotic promoter prediction. So for eukaryotic promoter prediction, again based on the presence of the unique feature of the eukaryotic promoter, that is presence of what? CpG islands. Okay, I hope you know this. In molecular biology you might have studied it. Okay, so here again GC reach, you remember we had discussed same kind of stuff. Okay, next so many vertebrate genes are characterized by the high density of CG, that means so much content, GC rich content will be there, dinucleotide near the promoter region, which is overlapping the TSS, okay, transcription sites. Okay, next, by identifying the CpG island promoter regions that can be easily traced on the upstream region of the islands. So once you identify this particular island promoter regions where they are, then you can easily identify the region of the island. That means you are having talking about the CpG islands, then just near to that particular region. Okay, so accuracy of the prediction can be improved by combining the CpG islands and other promoter signals, because we know that just behind these signals, just above, what you can say, that's above the signals, the promoters will be there. Understanding? So based on that indirect approach, we are going to identify or predict. So several programs are developed For the combined features, we are having CPG prod, MC promoter, compro, all these three things are there. We will discuss. CPG prod, web-based program. It predicts promoter containing high density of the CPG islands in mammalian genome structures or sequences, which we discussed in the introduction. It calculates average, what is the percentage of GC and as well as CPG ratio, that is observed divided by expected. That is the percentage. That is what you will get as a percentage. So, over a window of certain size, usually 200 base pairs. It will look for 200 base pair sequence. Next, when the values are above the threshold, then definitely the region is identified as CPG island, because above threshold means more rich, GC rich, what we call. Next, MC promoter, web-based program. Again, it uses neural network to make predictions. Neural network is one of the very complicated network in machine learning, and MC promoter uses this tool, uses this neural network. It has a unique promoter model containing 6 scoring segments. So it has got a particular model which has got a 6 scoring segments. It scans a window of 300 base pairs. There it was 200, here it is 300 bases for likelihood for being in each coding, non-coding promoter regions. So it checks for both coding, non-coding and as well as promoter regions. Okay. Next, input sequence physical properties such as DNA bendability. See, it also goes for signals such as Tata box, initiator box, CPG islands and all. Okay, these are the sequences or the information you can get. Next, put when you start, then the program combines all the features to derive an overall likelihood of for a particular site being a promoter. So when you can identify that promoter like that. Next, the program is currently trained for Drosophila and human sequences as a part of research. Next, finally, we are having the compro. It is again a web-based program. It uses a consensus method to identify the promoter elements for human DNA. Like if you remember phylogenetic analysis, we had done neighbor joining method, JAPNIF method, all those things and all. They all were based on the consensus method. Okay, like aggregated method. Like input will be here again cDNA, that is transcript sequence of gene. Next, programs uses information to search the human gene databases for the position of the gene, where exactly the position of the gene is there, that has to be searched. Now that's why it uses gen scan. Okay, it predicts 5' untranslated exons in the upstream region. So once the 5' most exon is located, that means that gene position of the gene is located, a further upstream region is used for the promoter prediction, which relies on the combination of 5 promoter prediction programs. There is TSSG, TSSW, we are having NNPP, ProScan and PromFD. Okay, no need to worry about the expansion. You just have to mention 1 or 2 is enough. For each program, the highest score prediction is taken as the promoter in the region. Definitely highest score will be taken. Next, if three predictions fall within 100 base pair regions, this is considered as consensus prediction. If no three-way consensus is achieved, then you can go for it will be TSSG or PromFD predictions are taken. So that is why consensus prediction it has to fall within 100. If at all it is going more than that, three-way consensus is achieved, then what you can for TSSG or PromFD predictions you are going to consider. Okay, so compro, that was about the finder, what you call promoter. Okay, for the eukaryote and eukaryotic also we discussed. Next, prediction of transcription factor binding sites, the third one. So here genes with the similar expression profiles are considered as co-expressed due to the common promoter and the regulatory elements. This one you already know what is co-expressed, right? Because co-expressed we call because of the common promoter and regulatory elements. Now the upstream sequences of the co-expressed genes that can be aligned together to reveal the common regulatory element recognizable by specific transcription factors. Now our idea is to find this co-expressed common promoters. Now this approach is experimentally based and appears to be more robust finding transcription factor binding sites. Like we are having meme, we are having melina, inclusive, phylocon and all. Meme, it's nothing but EM based program for protein motif discovery, and then also be used in DNA motif discovery. So melina is motif elucidator in nucleotide sequence assembly, that is the full form of melina. So it is again a web based program. It runs 4 individual motif finding algorithms simultaneously, that is MAME, Gibbs sampling, consensus and core search. So user usually compares the results to determine the consensus of motifs predicted by all the 4 prediction methods, and then he will come to a conclusion. Now next, inclusive, it's again a web based tool designed to streamline the process of microarray data collection and the sequence motif detection. 2 things: microarray data collection we want, sequence motif detection we want. Sequence motif means what? It's about again the short pieces of the proteins, and when microarray data collection, when you speak, it includes all possible information regarding introns, exons and the coding regions, non-coding regions, all those things. So the pipeline, how it starts: processes microarray data, it gets the data first, next automatically cluster all the data with based on the genes, that is cluster genes according to the expression patterns. Next, it retrieve upstream sequence of the co-regulated genes. It will take out only the co-regulated genes and then detects motifs using Gibbs sampling approach. So now what happens after that? So each sequence data is submitted to motif sampler 10 times. So results may vary in each run, okay, but the results from 10 runs are compiled to derive consensus motifs. Out of 10, if we are getting 3 results are common, okay, 4 results are common, and other 2 are having different values, the majority will be taken. Next, phylocon, that is phylogenetic consensus, Unix program to identify regulatory motifs by combining phylogenetic footprints with gene expression profiling analysis. So phylogenetic analysis, all of you know, it is nothing but finding the common ancestor. Here also the combining phylogenetic footprints with gene expression profiling analysis. With respect to the gene expression, what is the combination, what is the connection to a particular sequences that you are going to identify. Next, phylogenetic footprinting, that's why phylocon it is, consensus, phylogenetic consensus. So phylogenetic footprinting, identification of conserved, non-conserved DNA elements having a crucial functional roles. How? It takes advantages of conservation among the orthologous genes and conservation among the co-regulated genes. So once you have all this information, it is very easy to predict. For each individual gene is in a set of co-regulated genes, multiple sequence homologs are aligned to derive profiles. Multiple sequence homologs, all of you know, again it's nothing but multiple sequence alignment kind of stuff. So you can compare. So based on the gene expression data, whatever the information you get, these profiles are further compared to identify functionally conserved motifs. Now again you are going to compare especially to identify the conserved motifs among the evolutionary conserved motifs. So once you get that, definitely you can find the that gene expression profiling, okay, with respect to the phylogenetic analysis. So next, final one is predicting the translation initiation sites. So translation is definitely the key process for gene expression. Only after the translation we can say that gene is expressed. So in eukaryotes, what happens if the translation this sites, okay, is known, then there is no intron interrupt the 5' untranslated regions, or you can say this, you can say Kozak's rule probably will locate the correct initiation codon in most cases. But in what happens in prokaryotes, splicing is absent. Splicing won't be there, RNA splicing won't be there. So but finding initiation codon within ORF is still difficult because of the existence of polycystonic operons. Many operons will be there. So the open reading, identifying the initiation codon within the one ORF is difficult in prokaryotes compared to the eukaryotes. So that's that is the reason we are going that is one of the what you call sorry, one of the drawback what we can call. So tools are there, titer is there, ADG PR is there, TIS hunter is there, try TISA is there. So titer, titer, or you can say it predicts based on the TIS, that is translation initiation sites, okay, and it's based on the available high through output sequence, that is STS data. All of you know available sequence data is different with STS. STS, when you speak about high through output sequencing data, it's more accurate kind of data. So it is a deep learning based framework that allows to model the sequence features of translating initiation and identifying the potential transcription initiation site. So it can also identify the significant sequence motifs for different transcription initiation site codons. So it is a desktop based program, that means you can download and then you can install it and then you can use it. Next, ATG-PR, it's a program for identifying again initiation codons in cDNA sequence, specifically designed for cDNA, and it can be used to predict whether the initiation codon is present or absent, okay, in a piece of cDNA. You take a cDNA, it will able to predict whether the initiation codon is there or not, which ATG is the initiation codon for the cases when the codon appears to be present. If the codon is present, then your result will be ATG will be present. So web based, again this is a web based program. TIS hunter, a highly accurate predictor for TIS in human mRNA. Now all of you know what is TIS, transcription initiation site. So web based program again make use of edit kernels, that is which are simple but significant biological and probabilistic interpretations. It converts the region of the input mRNA sequence downstream to a putative TIS into amino acid sequences, and then to avoid high redundancy in the genetic code. So whatever the information you are having with respect to the mRNA, that has to be converted into the amino acid. So that is what is happening here in order to reduce the redundancy. So now try TISA. Try TISA, here again it goes for the accurate prediction of the TIS of microbial genomes, specifically microbial genome. For good predictions of a TIS, the number of TIS it has to be related features to the length of the regulatory signal, start codon usage, operon structure, and coding potential. These are 4 factors it depends on as having invoked in the design of the prediction methods. Now it classifies all the candidates into 3 categories. What are those 3 categories to the Darwinian evolution? That is true TIS, false TIS upstream of the true TIS, and then false TIS downstream of the true TIS. It's like Darwin evolution, how you told false, true, false, okay, like that it is. Now this tri-category of candidate TIS is a universal property of for the all the genomes. Now hence this method or a principle is applicable for all the genes. Since it is depending on 3 categories, we are calling it as tri-TISA. One is true TIS, false TIS upstream of true TIS, another one is just vice versa. Here it is false TIS downstream of true TIS, upstream and downstream, okay. That was about tri-TISA. So next topic is integrated gene parsing, that we will discuss in the next class. So we have discussed the 4 major topics of the concept detecting functional sites in the DNA. They can ask for entire question for 10 marks or 14 marks or 12 marks something, or they might ask 1 or 2 things from this topic. So one major topic is done, another 2 major topics are there. I will be making 2 more videos.